All right, we will be looking at the practice question in the notes for epistasis. Okay, so the first question is the type of inheritance shown by the two genes, right? So as the question says, one gene, gene B, determines whether there is the presence of bands. The other gene, gene M, determines the bending pattern, right? Whether it's mid-banded or five-banded. Okay, so in this case, it is epistasis, or if you want to be more specific, dominant epistasis. Okay, but just to take note, right, when questions in exams especially, like ask you to identify the type of inheritance for epistasis, right, or the kind of gene interaction, right, all you need to mention is epistasis, because again, you are not required to memorize the different examples of epistasis. Okay, so you just have to say epistasis. Okay, of course, if you are very confident, right, that it is dominant epistasis, you can give dominant epistasis. Okay, the danger is this, right? So if it is supposed to be dominant epistasis, right, you will be correct if you just say epistasis. But if you say recessive epistasis, you get wrong. Okay, so we cannot like ignore the recessive and mark you correct because you said epistasis. Okay, so again, if you are very confident, I encourage you, right, to be a bit more specific. But if you are not that confident, then if you want to play safe, then all you need to mention is epistasis if the inheritance is really epistasis. Okay? Okay, anyway, why is it dominant epistasis, right? Then that would be the answer to the next question, right? To explain how the big B allele, right, affects gene M. Okay, so the big B allele is epistatic. It masks the expression of gene M because it gives unbanded shell, right? So when the shell is unbanded, then regardless of the genotype of gene M, it will be unbanded, right? So there's no bending pattern per se when there is no bands, right? And so that's why the gene B, rather the dominant genotype of gene B, right, masks the expression of gene M. So because it's dominant genotype of gene B that masks gene M, so it's dominant epistasis, going back to part 1. Okay, so you're supposed to draw the diagram next, and this would be the diagram that you should have drawn. Okay, so again, it's kind of like a normal dihybrid diagram. Okay, the question told you, right, that the unbanded snail is double homozygous dominant, crossed with a snail with 5 banded shells, so the only genotype possible is homozygous, double homozygous recessive. So the F1 are double heterozygotes, which are interbred, right? So the siblings interbreed, which should give you the 9:3:3:1 ratio, but because of the dominant epistasis, it becomes 12:3:1 instead. Okay. Next, right, it goes back to the autosomal linkage that we have looked at in the previous lecture. Okay, so the term linkage means that two genes, right, are found on the same chromosome. So in the context of the question, the two genes are gene B as well as the gene coding for the color of the shell, right? They are found on the same chromosome. Right? So therefore, they are not, they're not inherited independently. There's no independent assortment between the two genes. Okay, so if the question, because there's no marks given for the question, right? So that's why it's a very short answer here. But if there are more marks given, then you can mention the lack of independent assortment, right? All these are features of linkage, right? So you can mention all those as well. Okay, but the gist is that the two genes are found on the same chromosome, right? So anyway, going to the next part, expected phenotypic ratio for the offspring, it would be 1 unbanded pink to 1 banded yellow, right? So just to explain, this is the diagram that you would have drawn if you are asked to draw. Okay, the question states that the gene B, right, is tightly linked to the gene coding for color, right? Tightly linked, right? So therefore, there is no crossing over between gene B and gene P. Okay, so no crossing over between the two genes. So you see there's only the parental gametes and no recombinants. And so therefore only 2 phenotypes instead of 4, right? So only the parental phenotypes, right? No recombinant. Right? So therefore it's 1 unbanded pink with 1 banded yellow, which are the parental phenotypes. Okay? Right, so that would be the end of epistasis.